Have you ever stopped to think about how we create some of our most powerful, life-saving medicines? Well, a huge piece of that puzzle lies in a technology that has, kind of quietly, revolutionized modern biology. We're talking about everything from vaccines to cancer therapy, and it all comes down to the incredible science of growing living cells outside of the body, creating this tiny living world right inside a laboratory dish.
But let's start with a really important question. How do you develop and test a brand new drug or create a vaccine without immediately putting people or animals at risk? The answer, for the most part, is this amazing technique of cell culture. It's become our first line of defense, and honestly, one of our greatest tools for discovery.
So here's our game plan. We're going to break down what cell culture actually is, how scientists build a home for these cells, how a culture even gets started and is kept alive, and then the really cool part. We'll explore the incredible things this science makes possible. So, let's dive in.
Okay, first things first. What does it actually mean to grow cells outside of the body? Let's get a really clear definition on the table.
To put it simply, cell culture is the whole process of taking cells, whether they're from humans, animals, or even insects, and getting them to live and grow in a totally artificial lab environment. You can think of it like creating this custom-built home that gives a cell everything it could possibly need to not just survive, but to actually thrive and multiply.
So what does it take to build that perfect little cellular home? Well, it's a lot more than just a plastic dish. You have to meticulously, perfectly, recreate the conditions of a living body.
The best way to think about it is like you're building a 5-star hotel, but for cells. First, you need absolute sterility. No bacteria or fungus crashing the party. Then you need the nutrient medium, which is basically a gourmet all-you-can-eat buffet. The cells also need a suitable surface to attach to, and the environment, well, it has to be perfect. The temperature, the pH, the gases, all of it. And of course, you need your VIP guests, the cells themselves, and all the right equipment to run the whole hotel.
Let's look a little closer at that gourmet meal, the nutrient medium. Scientists have a pretty big choice to make here. On one hand, you've got media that contains animal serum. It's super complex, it's packed with growth factors, but it can also be really variable from one batch to the next, and it carries a risk of contamination. On the other hand, you have serum-free media. This stuff is chemically defined, which makes it incredibly consistent and way safer. The catch, you have to know exactly what your specific cells like to eat.
All right, so we've built the hotel, we've prepared the menu. Now, how do we actually get the cells from a living organism into our culture dish in the first place?
Turns out there are two main paths you can take. You can do what's called an organ culture, where you keep a tiny piece of tissue totally intact. This is fantastic if you want to study how different cell types talk to each other in their natural neighborhood. Or, and this is more common, you can use enzymes to gently break that tissue down into individual cells. This lets you grow a really uniform population, usually in a single flat layer on the dish, which we call a monolayer.
And this leads to a really important progression. So those first cells you get directly from the tissue, that's your primary culture. Then, when you take some of those cells and move them to a new dish to give them more room to grow, you're creating secondary cultures. If you keep doing this, you can eventually develop a really stable, uniform population called a cell line. And that becomes an incredibly powerful and consistent tool for all kinds of research.
Now, starting the culture, that's just the beginning. Remember, these are living, growing populations of cells, and managing them is this dynamic, ongoing process that needs constant attention.
Just like any population, cells in a dish follow a pretty predictable growth cycle. First, they're in the lag phase, just kind of getting used to their new home. Then they hit the log phase, and this is when you see rapid, exponential growth. They're just dividing like crazy. Eventually, though, they reach the plateau phase. Growth slows way down because the dish is getting crowded and the food is running low. A lot of normal cells also have this neat trick called contact inhibition. They can literally sense they're touching neighbors on all sides, and that's their signal to stop dividing.
So, what happens when the cells get too crowded? You can't just leave them packed in there. They need to move to a bigger apartment. And this really critical process is called passaging, or sometimes subculturing. It's absolutely essential for keeping the cell line healthy and growing.
For cells that actually stick to the dish, this means you have to perform a little procedure called trypsinization. First, you get rid of the old medium and give the cells a quick wash. Then, you add an enzyme called trypsin. It basically acts like a pair of molecular scissors, snipping the protein glue that holds the cells onto the surface. You let it sit for just a little bit, and boom, the cells detach and start floating. Finally, you add fresh medium to stop the trypsin from working, and you can collect all those floating cells and move them to their new home.
But here's a fascinating twist. Unlike immortal cancer cells that can divide forever, most of our normal cells, well, they can't. They have a built-in expiration date, and it's known as the Hayflick limit. After about 50 divisions, give or take, they enter the state of retirement called senescence and just stop dividing. This limit is fundamentally tied to how we age, and it's a huge challenge that scientists have to think about when they're working with normal cells.
So, we've gone through the what and the how. Now for the most important part, the why. Why go through all this trouble? Because the applications, this is where the science literally changes our world. Let's look at the miracles this little dish makes possible.
Let's start with viral vaccines. See, viruses are parasites. They need living cells to make copies of themselves. Cell culture gives us a safe, totally controlled factory for growing enormous quantities of a virus. This is the technology that's behind some of our most important vaccines, like for polio, rabies, and the flu shot you get every year. It's just so much more ethical, scalable, and safer than the old ways of doing things that relied on whole animals.
It's also absolutely essential for making many life-saving medicines. A lot of modern drugs are actually complex proteins, like antibodies, that are designed to hunt down and fight cancer. Simpler things like bacteria, they just can't build these properly. You need the sophisticated machinery that you only find inside an animal cell. And this is where CHO, that stands for Chinese hamster ovary cells, come in. They are the workhorses of the biotech industry. So the next time you hear about a powerful cancer drug like Keytruda, you can picture millions of these tiny CHO cell factories that made it all possible.
And if we look to the future, there's gene therapy. Cell culture is the fundamental workbench for this entire revolution. It lets scientists do amazing things, like take a patient's own cells out of their body, correct a genetic mistake right there in the lab, and then put the healthy, repaired cells back into the patient. This approach is an incredibly powerful platform for developing cures for diseases we once thought were completely untreatable. To put it bluntly, without cell culture, modern gene therapy just wouldn't exist. So, from understanding the absolute basics of biology to manufacturing the most advanced medicines on the planet, cell culture is this quiet, unsung hero of modern science. It's a world in a dish that has unlocked countless secrets. Which leaves us with one last powerful question to think about. Now that we have the power to grow and even manipulate the very building blocks of life, what kind of future will we choose to build with it?